Hey, what's up, you guys? I'm making this video, so hopefully this goes well in one take. I'm deciding to make this video because just to kind of clear up misconceptions and just kind of help you guys out. I told you I wouldn't do this, but I'm feeling that some of you guys may need it. So those of you guys, I already read a couple lab report introductions, first drafts, and they look excellent for those that I already read. So I think you guys will do fine. But just a quick rehash on the lab. I know it's our first lab, so I will do this for you guys. Um, so let's see. Introduction. So introduction again. Remember, I'm breaking it up into three parts. Uh, the introduction paragraph, and then the purpose, and then lastly the hypothesis. Usually, like I said again, the introduction has all three components, but I'm breaking it up just so that I can, for my sake, so I know like how you guys are researching. But I think it's going to help you guys also to really focus on the research aspect. So just the introduction paragraph is I have notes over here, the background information from your scientific sources. So the article that I gave you, which was this over here, this is one scientific source. It's amazing, right? You talk about Newtonian versus non-Newtonian fluids and viscosity. There's everything about shear, right? Really important for polymers. They even have diagrams that you can use, right? There's this article is just pure amazing, okay? Because it actually deals directly with slime. And for this lab, you are required to do two sources. You can do more if you want, but two. And one of it could be this article over here, which I highly recommend you use. So you do need to do some other research here and there. But slime is and polymer is such a wide talked about topic that I don't think you'll have problems with that. But background information from the scientific sources. So, and ease the reader in. Talk about polymers. Talk about slime. Talk about the chemistry. Right. Ease the students. Ease the reader in. Okay. You're going to be also explaining the problem or area being investigated. Okay. So the reader just can't. I mean, slime is pretty much straightforward. If you're gonna do an experiment with slime, it's about slime. But if you have a more broad, general topic, just by giving background information about that topic isn't really gonna talk about what you're actually trying to hone in onto. So explain the problem or the area being investigated. Okay. And also try to answer why is this even important. Okay. And not like why is it important to society, right? Sometimes yes, but why are we doing this? Why is this lab important? Okay. And then next, you're going to transition to the purpose. So the introduction is going to flow directly into the purpose, and the purpose, you can read this over here. Okay, I think this is very concise. I'm trying to make this video short. And lastly, you're going to transition into the hypothesis, right? Remember, after you write your hypothesis, the if and then statement, um, you have to explain why. Okay, and then that needs to be backed up by sources. You can't just say because I think so. Okay, that you're a scientist. You need to have some backup materials. Materials, I'm not going to cover too much. Procedure, if you already did your procedure, your first draft, and I know we use different grams of different things, right? You don't need to change that. I'm looking for just a general procedure and how you actually completed the lab. Yeah, I'm not looking, uh, I'm not gonna bug you over like having like 4 grams of water versus like 7 grams of water. On other labs that do require accuracy, though, I will expect to have very accurate procedures. For data results slash observations, this is pretty much where you tell me what is going on, okay, without any explanations. Just objectively tell me what is going on. Okay, so give me the results, no explanations. Now, if the lab is either just qualitative or quantitative, meaning you're only looking for quantitative data or only looking for qualitative data, you don't need to put two sections. But if you are doing qualitative and quantitative, just like for the slime lab, I do highly recommend, right, doing two sections. So for example, this formal lab report, this lab report has a quantitative results section and a qualitative results section. Makes it easier for the reader and it makes it much more organized for you. Now, I think for period two, I might have said you're required to do a graph. No, you're not required to do a graph. For this particular lab, if you think about any variable that we change, right? We're not doing multiple trials or anything. We're not looking for an outcome of just one variable. We have so many different things to talk about, right? Both quantitative and qualitative. So I'm not really looking for this graph over here, but I am looking for charts, okay? The table is not going to look like this for you guys because we have so many different types of quantitative data. For this is just talking about temperature versus time, only two variables. For us, we're talking about measurements, time it took for it to hang, other measurements. There's a whole bunch of different types of measurements you can take for quantitative data. So you guys will just have more of like a table of lists rather than like a table that actually shows that portrays a graph. Um, Qualitative results, you're gonna have I think you should have a sizable paragraph. There's a lot to talk about for slime. It's a lot that you've observed, right? Observations can go over here. And the next is calculations slash analysis. Now, there are no calculations for this particular lab because we don't have any, we're not trying to compare it to some kind of experimental value or theoretical value. But the analysis portion is to explain your results section, your data and results section. So this is why you don't explain anything in your data and results section because you do this in your analysis, okay? And then what do you call that? If you go up here, this analysis section has some detail. So whatever results you get, you're explaining. That's the main focus. Okay. Lastly, conclusion. I'm not going to go into the conclusions at all, pretty much, because of how many questions I give you, guiding questions, and how much material I have. But general purpose is you're going to answer the purpose, answer the objective. What I mean is you're going to tie everything in back to the objective. Okay, follow these guidelines, follow these questions that I'm asking you guys, and lastly, finish with sources of error. If you ever say human error, right? This experiment didn't go accurately because of human error. Just by saying human error, I'm not going to even give you credit for sources of error. First of all, you're required to have two, but you guys are in honors chemistry now, so you can have. I know you guys can do better than that. Okay, and that is about it. So this will be due on Monday. Okay, today's the 25th, so Monday would be, I might use this video for next year. So Monday, this coming Monday. All right, you guys don't need to take notes on this. It's already been 8 minutes, so I'm gonna be quiet now. All right, take care, y'all. Bye. Oh yeah. Lastly, you guys, one thing I wanted to talk about was for the data and results section. I quickly skipped this important piece. For for where's my mic? Ah, my mic's. What a waste of time. I don't know if you heard that, you guys, but lastly, I wanted to add in a little thing about the data and results for the slime lab in particular. For this slime lab, you are going to have quantitative slash qualitative data for the slime you made, which was today, the first day, and you're going to also have qualitative slash quantitative data for day number two, which you'll make tomorrow, the second day. So you are going to do a comparison, right? That comparison is going to help you answer your hypothesis or to either prove or disprove your hypothesis. Okay, so you are going to have sort of two sets. So when I make my data tables, my chart, I'd have some sort of organization where it's day one and day two, or maybe like the experimental trial, right? Experimental trial versus, whoa, not experimental trial, just the standard trial versus the experimental trial. All right, you guys, peace.